The production of A-beta takes place in all individuals and in many cell types, but the highest production of A-beta occurs in neurons in the brain. A-beta is derived from a parent protein called amyloid precursor protein, or APP, which is processed through two normally occurring pathways, often called the non-amyloidogenic pathway and the amyloidogenic pathway. The most prevalent pathway is the non-amyloidogenic pathway, in which APP is cleaved by the enzyme alpha secretase, producing a large fragment which is secreted into the extracellular space. In contrast, the amyloidogenic pathway is an alternative, less prevalent pathway for APP that does lead to A-beta generation. The initial proteolysis is mediated by a different enzyme, beta secretase or BASE1. This cleavage also results in the release of a large fragment into the extracellular space, but the remaining fragment starts with the first amino acid of A-beta. Subsequent cleavage of this fragment by gamma secretase produces A-beta peptides of different lengths. Most of the full-length A-beta peptide produced is 40 amino acids in length, A-beta 40, whereas a small proportion, normally about 10%, is the 42 amino acid variant A-beta 42. A-beta 42 is more problematic than A-beta 40 because it is more hydrophobic and more prone to aggregate. It may form groups of 2, 3, or 4, or even larger oligomers and eventually form fibrils and plaques.